Bahamas Ministry of Agriculture, Marine Resources, and Family Island Affairs, and its subsidiaries, BAIC, BAFSA, BAMC, and the Department of Agriculture. I am your moderator, Dr. Shelley Bridgewater, livestock scientist, country representative, CARDI, Bahamas. It's really a pleasure to have you with us. Of course, we would like to welcome all our registrants from the Commonwealth of the Bahamas, along with some of our regional brothers and sisters hailing from Trinidad and Tobago, Jamaica, Barbados, Grenada, St. Lucia, St. Kitts and Nevis, Antigua and Barbuda, Curacao, Belize, St. Vincent and the Grenadines, and Guyana. We would also like to recognize our international friends coming all the way from Nigeria, the Philippines, United Kingdom, and the United States. Welcome, welcome, welcome. We do apologize for the slight delay in our start, but now we do hope that at the end of this series, you would have learned more about sweet potato production. Just to give a brief background, CARDI, under the CARICOM Regional Transformation Program for Agriculture, has a lead responsibility for the development of the regional sweet potato industry. It is one of those versatile roots and tubers that has a high calorie value, and it is a rich source of vitamins and nutrients and lends itself to being prepared in a variety of ways. Not only this, it is an integral part of the Caribbean cuisine. We would briefly like to review some housekeeping rules before we start. All participants, your microphones will be muted throughout the presentation. Chat is also disabled for participants, so it means that attendees can chat with no one. All questions to the presenter will be addressed during the question and answer session immediately after the presentation. However, you are free to type and send your questions throughout the presentation using the Q&A tab available at the bottom of your screen. Simply type your question in the question and answer window and click send or enter. If you do not want your name to be displayed with your question, you may check send anonymously. The presenter will then answer your questions live via microphone so that all participants are able to hear. So before we introduce our presenter, we will kindly ask you to take 5 minutes, just 5 minutes, to answer the following 7 poll questions. If you go to the base of your screen, you may click on the poll icon, which will give you access to the poll questions. If you do not want your name to be displayed whilst responding to the poll questions, you can check send anonymously. So don't worry, it's not a major exam, but we're going to use this information to compare your knowledge base before and after training. So you may now go to your poll questions. Great. So your poll questions are presently up. So you can start answering those questions. You have approximately 5 minutes. So great. So most of you who have just joined in, right at this time, we are conducting a poll just to assess your knowledge prior to the training. And we will just ask you to click on the item polls, which is at the base of your screen, and it would allow you to see the 7 poll questions. You can answer those questions and submit it. In addition, if you choose to be anonymous, you can actually select send anonymously. Good, so your 5 minutes are now up. So as you can see, it was not so bad. So your answers will actually be reviewed at the end of the session. So without further delay, we would like to warmly introduce the presenter, Dr. Annika Minott, plant health specialist scientist at the Caribbean Agricultural Research and Development Institute. She also serves as a CARDI representative for the Cayman Islands. Her educational training is centered on a multidisciplinary approach to plant health and includes entomology, plant pathology, nematology, plant and soil science. Her research work interest is related to crop science, roots and tubers, and integrated pest management. So we will now turn over the presentation to Dr. Annika Minott.
Thank you, Chair. I'll just go ahead and share my screen. Hi, good morning, everyone. Pleasant good morning. Thank you all for attending. I will be presenting some general tips for sweet potato production. Without any further delays, I will just get right into it. Okay, so for sweet potato, the Latin name is Ipomea batatas, and it's a root crop. So oftentimes we say tuber, we mix between tuber and root crop, but it's really a root crop. We have lots of varieties. They come in various shape, sizes. You have different colored skin, different colored flesh. The leaves and the roots, they're edible. They're a good source of vitamins A, B, C, and E. It also have some amounts of iron, zinc, copper. The sweet potato is also a good source of animal feed as well. And it has use in the industrial products such as starch, alcohol, distilled beer. So it's really a perennial, which is grown as an annual, meaning that you can plant it and it keeps on having cycles of vegetative production, then going into the root production. But because of pest and disease issues, it is commonly grown as an annual. So it's really better to plant, have one cycle, remove that from the field, and then go again, right? So it needs moderately warm climate. It does not tend to do well in shade, in too much shade, as that will reduce yield. And it can tolerate drought conditions, but it does not do well in waterlogged conditions. And sweet potatoes, they can be harvested anytime between 2 to 6 months after planting, depending on the variety. So just a look at some of the nutrient value for sweet potato. So in the raw form, we're seeing high energy. This is for the roots. So 105 kilocalories or 439 kilojoules compared to when it's baked in the skin, still having similar value also when it's boiled without the skin. If we look at dietary fiber, we're seeing 3 grams, both in the raw form and baked in skin. When it's boiled without the skin, we're seeing a reduction to 1.8. Calcium values also high. Phosphorus, potassium, vitamin C, and see your vitamin A's, right? So we're seeing the nutrient value of the edible portion of the roots, right? Okay, so let's have a look at the plant, the parts of the plant. So on the right here, an illustration of the sweet potato plant from the International Potato Center. This is SIP, based in Peru, and they do work on sweet potatoes, and some other root crops. So we see that we have our apical shoot here. In the top portion, we have our apical shoots, we have our leaves, we have flowers. And the sweet potato, it tends to be viney or upright or, spreading a bit. So you have different forms to it. And below ground, we have our storage roots, which is what we'll eat. We have our fibrous roots, which will be responsible for taking up the necessary nutrition, the necessary ions and stuff that will feed into the plant. And we also have our pencil roots. So these are elongated, they are more hardened versions of the fibrous root, right? And these will go down, if it's grown as a perennial, these will go down and possibly bulk up, like somewhere along the line, bulk up to get storage root. The flowers and the seed, they contribute to the production of new varieties. When we plant sweet potato commercially, what we're essentially doing is getting a clone of the parent by using the vegetative portion. So when we cut the slips, we're actually getting a clone of the parent material. So this photo here, we're looking at, so the plant and just a real version of what is here without the flowers. So this part here, the seeds, we don't normally grow the seeds unless it's for breeding and you want to improve the plants, right? And I have a photo here of a node, which are the points where the leaf connects to the vine. And what is cool about the sweet potato is that every time these nodes get in contact with soil or any growing media, they have the potential to send out roots. And this is what we take advantage of when we grow sweet potato. We take advantage of these nodes, right? And these nodes, so essentially what the sweet potato is doing, it's harvesting the sunlight and converting that to energy. And so the energy, you use that to form the different parts of the soil and so forth. But then some of those sugars, it's stored into the roots, right? Which is what we eat. And you'll have a back and forth exchange, a sourcing relationship where sugars are moving back and forth. So there's a period in the sweet potato where it's storing sugars, and there's a period when it's sending it to produce more leaves or that sort of thing. That's after it's established.
So sweet potatoes, to get a new plant, you can either use vine cuttings or the tubers. So what I'm going to demonstrate is selection of the vine cuttings. You want to take the cuttings from the tip of the vine. You do not want to take any portion that is close to the roots, right? So you're going to take from the tip of the vines, and you're going to look for between 12 to around 14 or 16 nodes, and the nodes are these points of connection to the leaf stem and the main vine, right? So here we have, so these are on opposites. In the case of this vine, they're on opposite sides pretty much. So we won't start with here. So to count, we'll do 2, 4, 6, 8, 10. So I'm gonna do 10 nodes. Sorry. Right? So from the tip. And this you're going to treat with fungicide, insecticide dip, right? To get rid of any eggs or any insect or so that is that might be on the slip. So this is what you'd call a slip, and this is what we're going to plant.
Okay, so I'm just trying to make this as interactive and practical as possible. So just a little video to demonstrate how to select the vines. We always go for the tip of the vines because that decreases the chance of having silas or any of those root pests present. There's also a technique where you can use 2 nodes to multiply the amount of cuttings you can get. So the options for planting material, pretty much tissue culture plantlets. And if you have tissue culture plantlets, that is always the best option. Then we have vine cuttings, which I just demonstrated, and or also allowing the roots to sprout. So you want to select the vine cuttings about 20 to 45 centimeters in length, tip free at the tip, as I indicated, and you treat those cuttings with insecticide, fungicide dip. You can plant those 30 centimeters apart or more, and you want to ensure that during the first 2 weeks, this is well watered. So sweet potato, they can be grown any time of the year. And depending on your location globally, it seems to do well when it's a bit warm and dry. But this can also change based on where you are and your soil and what is there as well. So, but it can be grown any time of the year. So land preparation, it can be grown on marginal land, but if you can, you get a better deal by selecting land that is well drained, sandy or sandy loams. Silty or clay loams as well could be fine. You want a place where there's no history of sweet potato for the last 2 years. Ideally, you don't want saline or alkaline soil. I know that may be an issue in the case of the Bahamas. But in general, though, we want to do any kind of fertilization based on a soil test, right? So for sweet potato, we want to use individual mounds or ridges. You can do flatbeds, but if you have a hard pan or if it's very clay, that will be a problem in terms of misshaping the roots when they're bulking up or restricting them. I personally prefer mounds and ridges because it helps with drainage. It's helpful for the vine turning exercise, which is very important. It's a very important husbandry activity when growing sweet potatoes.
So it's a good idea when you're preparing the land to plant sweet potato and pretty much any other crop that you basically till the soil, and you could either till the area that you're going to plant and leave the other areas that you're not planting without being tilled, or you till every part of the section that you're going to plant, right? We also recommend that you plant sweet potato either in individual mounds or in ridges. Now, if you have the land space, this is ideal because you can go ahead and turn the vines easier more than doing it on the mounds. So on the mounds, we want to at least have about 40 centimeter wide base and a minimum of 20 centimeter height, right? And this is important for those varieties that go deeper because you're going to give the tubers, you're going to give the roots more space to grow before they hit the hard part. And having the mound or the ridges, it also helps you to harvest the roots easily when they are mature. So if we're gonna do a mound, you just go ahead and you can use a spade or a machete. You make a hole here so you kind of make it deep, and you can go ahead and either you do vertical or horizontal. What I did, I removed the leaves from the slip. You don't have to remove them. I just did that because the sweet potato is going to shed those older leaves while it's growing. So I'm gonna go ahead and do this horizontally, and you want to get as many nodes as possible into the hole, and you go ahead and cover. For the mold, if you're going to use drip irrigation, it's a good idea to plant your slips next to the emitters. And for sweet potato, you can use a minimum of 1 foot spacing apart, right? right? So you can go ahead and punch your drip tape and place the emitters 1 foot apart or ruler length apart. Right, so we'll go ahead. I'm gonna go ahead and put vertically. You want to cover as many notes as possible. So, right. Go ahead and do this horizontally.
So the orientation of the slip, it really doesn't matter. You can do vertically, you can do horizontally. For the backyard gardeners, I know space can be a limitation, so you have the option of, using these bags, tires, and the tires can even be arranged differently. Buckets. In this case, this person cut the bottom of the bucket and turn it upside down, and you have other possibilities, grow boxes, etc. So all right, let's have a look at weeds, right? Simply put, and in the most general context, weeds are plants that grow where they're not needed. And being plants, they compete with crops for water, light, nutrients, and space. And they also may serve as alternate hosts for diseases and pests. And this will affect the crops growing. Those diseases and pests can affect the crops growing, right?
So it's a good idea before you do any planting that you till the soil, you go in and till the soil. You could do a minimum tillage where you're only tilling the area that you're going to plant, or you till the entire area that you wish to plant. And what this does is it incorporates air into the vicinity. Some of the
Right, so by tilling the soil, and as I said, you can do minimum tillage or you till the entire area, you're introducing air, you're basically overturning the soil. So those oh, my screen is not being shared, right? No, sorry about that. I don't know what happened. Oh, it is, okay. My apologies. Right, so the video earlier was saying that, it's good to till the area. You can either till only the area that you're planting or the entire area. And this allows you to incorporate air into the soil, and it also exposes weed seeds that are buried below. And those weed seeds, when they come to the top, they germinate and, well, they germinate, sprout, and then you get a chance to get rid of those in the young stage. So weed management, right? It's good to look at preventative and reductive measures. This starts from before you even plant. You want to monitor the area for weeds. You want to get rid of those weeds from the early stage before the sweet potato covers over the soil. And once the sweet potato covers over the area that you planted, it is pretty fine because it will compete with the weeds present. So you can use the machete, weed whacker. I don't really like to use the weed whacker though, because I find it basically cuts the top off of the plant, of the weeds, and then you are left with a mat basically, and after a while, you know, that area gets compacted. You can also use herbicides as well, but we have to be mindful that sweet potato would be a broadleaf, so you won't be able to use herbicide that affects broadleaf. So for sweet potato, competition between crop and weeds, and this is applicable for a lot of other crops in the early stages. So the competition between the crop and the weeds in the early stages will affect yield. So the critical period for managing weeds with sweet potato is anywhere between 4 to 6 weeks after planting. And this can start as early as even 2 weeks after planting. So this is a good time to target weed management. At 6 weeks, the sweet potato will basically cover over. If you're having different rows of it, it will cover over, forming a canopy. And you also want to go in beforehand and till, as I mentioned, allow those to sprout, and then you go in and get rid of those. Some places, and what I just described is a stale seed bed technique. You also can use soil solarization, where you cover over the area with clear plastic mulch or black plastic mulch, allow the soil to heat up. And by that, it will heat up to a certain temperature, and those will kill weed seeds or any fungal spores in soil. And as I mentioned about the chemical intervention as well. So let's have a look at a few examples of weeds across the nation. So we have Amaranthus species, and this is the one here on the top right of the screen. It's related to in Trinidad, Baji, in Jamaica, Callaloo. I think in America, they call it pigweed. So this here, amaranth, this is one of the weeds that we look out for. It's a known host for the white rust in sweet potato, so that's one that you'll be on the lookout for in terms of removing. You want to look at whether you have a lot of broadleaf weeds or grass. So broadleaf just mean that it's not a grass and the leaves are broad. Just a general term. You want to look at whether the weeds are, so look at this weed here. This is this grass weed here, right? It's erect and it has a potential to be taller than the sweet potato. So if you have a lot of that growing with the sweet potato, it can basically shade out, or even this Amaranthus species here. If you have a lot where the sweet potato is growing, what it might do is shoot higher, go up higher than the sweet potato, and can crowd out and block out the sunlight. At, you know, how the weeds grow versus how the sweet potato grow, and you determine in terms of getting rid of them. The purslane here, watergrass and purslane. So watergrass is this one, the far left of the screen at the top, and the purslane is the far right, lower right. So these, you don't want to use a weed whacker or a brush cutter with this, because the small pieces that you leave in the field, they can act as vegetative propagators. So they're like sweet potato, where they have nodes and so forth that will form a new plant. So you're spreading it by using weed whacker. So the type of weed will also influence what kind of action you take to reduce the population, right? And this vining weed here, which is Meremia umbellata, which is the hog vine. Because it's a vining weed, you need to find the root. You can't just cut off the top because you basically just cut off the top. It's still going to be there. So you have to eliminate it at the root, right? And some weeds, for example, the sedges, you have to get rid of the rhizomes as well, because if you just cut the top off, then below it will sprout again, right? So we have to look at all of those characteristics when we're looking at weed control. Another thing to look at is the white top, which is this one, below center, sorry for my descriptions. But this has reported some resistance to glyphosate. So that's something that we have to be mindful of. And this one here in the middle at the top is morning glory, which is related to sweet potato. So that means it also has similar hosts, similar pests to the sweet potato, and we don't want this in the field, right? So let's look at some scenarios here. So on the left of the screen, we see in here, this is a grass weed. It has started to flower, and we want to remove this weed before it gets to flowering stage, right? Left here on center left, this is the white top. We also see that it's flowering. We want to remove this before it gets to this stage, right? Here at the grass, this has not started to flower, and this is the stage we want to try to remove it at, right? And here I mentioned about sedges. So these have rhizomes. So if you're to pull this out of the ground, you'll see a little nut-like. That's why they call it nut grass. But if you just go in and cut off the tops of these, it will eventually sprout sometime after, because there is still a portion of it that will sprout, right? So we want to remove these from the root, right? Okay, so let's have a look at plant nutrient availability. So, soil, right? Soil, an important part of soil is the pH, right? And percent hydrogen, or the amount of hydrogen ions and the amount of hydroxide ions, right? Now, if we look here, we see that, for example, nitrogen is available between 6 and 7.5 pH. Iron, it's more available in the acidic side, which is, between 4 and 6.5. So basically, different nutrients in the soil, they're more available depending on if it's more acidic, which is over on the left between 4 and 7, or if it's alkaline between 7 and all the way up to 10, right? And this is important in the case of Bahamas and in other places, because some places the characteristic soil pH is that it's alkaline, and some places it's acidic. And because of that, you have to try and correct, or you have to adjust the soil for it to be able to grow plants, so you can grow it successfully, right? Because otherwise you would be putting the plant in an environment where it's, even though you're applying the fertilizer, it's not able to take it up, because the fertilizer or the ion, the nutrient that it needs to take up through the roots, it is not available in the soil, right? So, all right, let's look at it further, right? So for sweet potato, according to the literature, anywhere between 5.5, 5.8 to 6.5, that's the pH that we want for sweet potato, right? So this is that in this section here in red. Now we saw earlier that some of the nutrients, they're not available on this side, but they're available on that side. And some they're available on this side, but they're not on that side. So in the case of the Bahamas, most of your soil is over on the alkaline side, right? And the pH tends to be between 7.5 to 8.5, right? So we have to do a process of acidifying the soil, lowering the pH to allow for plant growth, right? Now, the good thing about that is that sometimes just by applying certain fertilizer, it is in fact lowering the pH. There's also, so you're seeing here, so causes of acidity here, rainfall, acidic rain, fertilizer, weathering, oxidation. Causes of alkalinity can be drought, weathering, high concentration of carbonates. Now, in the case of Bahamas, because of your limestone soil, you have a high concentration of carbonates, and the presence of that now allows for it to be an alkaline soil. So you tend to have iron and zinc deficiency, and you have an imbalance of calcium, magnesium, potassium. So we have to make sure that we're applying fertilizer that will bring the pH to a suitable range, and therefore allow for the plants to be able to take up these required nutrients, right? Now, in other cases, this in other places in the region, the soil is acidic, and what they'll have to do now is do a liming activity to kind of bring the pH up to a suitable range. So in Bahamas, it's the opposite that you have to do. So this can be achieved by applying elemental sulfur, but that will be the last resort, because by even applying some fertilizers, you're actually acidifying the soil. So that includes like ammonium sulfate, and even having ammonia there as well will help to decrease the pH a bit, right? So it's not always necessary that you have to apply sulfur to bring down the pH. Oh, and I'll touch on that a little further, right? So the next thing we're looking at is irrigation. We want to set up irrigation where possible. Ensure that during the first 2 weeks that the plants are well watered. And we have to be careful that we're not just watering the top layer of the soil, because depending on the irrigation that is used, for example, sometimes you're doing an overhead watering or a sprinkler system, it's going on the surface, but then depending on the soil, it possibly is just running off because of how it's hitting the surface. And what we see is that the drip irrigation, because it is there and it's slowly releasing the water droplets, it allows for the water to actually percolate and seep down into the soil, right? So by having it on for a while, you're actually wetting the soil, and over time you might see, depending on the soil particles, it might go a bit deep or go a bit wide. So that drip allows you to conserve the water, because the sprinkler, it's going there, there's a wind taking some, there's evapotranspiration, but you're having a less of that water loss happening with the drip, because it's a slow drip that spreads out. It goes down and out and keeps on doing that till they have a wider balloon, right? And although sweet potatoes can tolerate drought conditions, if you provide it with enough water, you'll get higher yield. It is always better to provide small amounts of water at regular intervals than a lot of water at once. As I mentioned before, you can just decide to just turn on the water for like 1 hour, but a lot of that is runoff because of how you're applying it, right? And so and what you're actually doing by doing that is the plant is there getting that. And another thing, the plant will only take what it needs. So it's you're giving that to the plant, but then that dries out and then you don't water it for a long time after. What the plant is going to do is adjust to that. And so sometimes because of that, now the leaves looking all droopy and it's not functioning as how it's supposed to function. And then, you know, the tubers come out cracked, you know, they split because they're adjusting the pressure and what has been going on. We want to also water plants either early morning or late evening hours. And this is especially because of the whole evapotranspiration aspect where, you know, with the sun shining on the water as it is released, a lot of it is lost through transpiration. So you want to water at those times. Also, some plants, when it comes to midday, they're not taking up the water as they should because they're also trying to focus on cooling down in terms of opening and closing the stomates. So it's the plant, for the plant, it just uses the water better in the early and the late time as well, right?
So earlier I mentioned about turning the vines, and this is a very important husbandry activity. And essentially, when you're turning the vine, what you're doing is you're discouraging the sweet potato from sending down any other storage roots, right? So in this case, I planted the sweet potato in this section, and I do not want it to send down roots anywhere else. So I'll have to go in, lift up the vines, pile it on top of each other, do the same here, pile it onto each other, right? And we'll talk a little bit about the nodes, right? So I mentioned earlier about the nodes, right? And the nodes are where the leaf stalk meets the main stem. Now, each node has the potential to produce roots, right? And the roots that we eat, they're actually storage roots. So if you allow the vines to run all over the place, what will eventually happen is that these nodes, some of these nodes will penetrate the soil and store the sugars, and then you'll have roots. And then when you are ready to harvest, even though you could have planted the sweet potato here, there are other sweet potatoes in other sections that you have to look for. So by doing the vine turning and being intentional about where you plant, when you're ready to harvest, you basically go to that root, and that is where you harvest, because this is, we're promoting storage of the sugars here. So this is where we'll go. An important activity is that of doing fertilizer application. And for the fertilizer application, what we want to do is we're going to find where the area you planted, and you basically have a little trench. You can do it on either side of the plant, and the applicable amount, so in this case I'm using 12/8/28. It's an all-purpose available locally where I am at, and you put in and cover, right? And we do it on the other section here. And by covering, what you're doing is reducing the amount of the fertilizer that is lost by volatilization, because, for example, ammonia contact with the sun will allow a lot of it to be lost into the atmosphere, and your plant won't get that, and then you'll be wasting the fertilizer and essentially your money.
Right, so I'm just gonna repeat a little bit. So I was demonstrating fertilizing. It's always a good idea to cover over the fertilizer. So apply it in a trench, cover where you applied, as opposed to just scattering it on the surface, because what will happen is that the nitrogen, for example, will volatilize, and that means it's just lost as nitrogen gas to the atmosphere, or ammonia gas to the atmosphere. And basically you could have used that, the plant could have used that to make food, but it's lost because it's exposed to the sun. Also, in terms of, remember with the nodes, everywhere those roots touch, they have the potential to form those storage roots. Everywhere where the node touches the ground, they have potential to form the storage roots. So, and if it stays there long enough, you might have tubers forming. So we have to go in and turn the vines occasionally so that it's discouraging the plants from sending down those roots to different places. And by turning the vine, you're basically forcing the plant to only store sugar and have those roots bulking up in the area that you plant, so that when you're ready to harvest, you go to that area and then you harvest. Otherwise, if you just allow it to go all over the place, what will happen is that when you're ready to dig, like, yes, you'll go to where you planted, but there are also going to be other areas where tubers will be available. And there's no way of really knowing where that is sometimes, right? So fertilizer, always, if possible, do it based on soil tests. I know that is something that's limited in the region, but is very important in terms of cost savings, in terms of, applying enough rather than over applying or even under applying. And, some of the problems we have, it can be fixed because we have that information to tell us, okay, your soil is acidic, and maybe by just, adjusting the pH, it will do better, that kind of thing. So having the soil test really gives you an understanding of what is going on in the soil, right? And just to say that too, so here we're saying that the NPK, the nitrogen, phosphorus, and potassium, it should be based on what is removed from the soil by the sweet potato. So basically, when you're applying fertilizer, it's to put it back to ensure that the plant gets, that new crop gets what it needs, right? So the rule of thumb, the nitrogen is at, well, potassium is at least 1.3 to a little bit over 2 times the amount of nitrogen. So here we see that according to these studies, nitrogen removed between 70 to 100 kilograms per hectare of nitrogen, and it also removed 100 to 210 kilograms per hectare of nitrogen, right? So we want to mimic that with our fertilizer. And we want to apply the fertilizer between the first 6 to 8 weeks. For those varieties that get ready in 2 or 3 months, we want to do that earlier. We also want to look out for boron deficiency in alkaline soils, and that would be applicable to the Bahamas, right? So just a look at this fertilizer. So this is a fertilizer I used in the video earlier, and we see in the total N is 12%. The available phosphate, which is, so your N, your P, 8%, and the potassium, which is the K, 28. So this is a 12, 8, 28. We're also seeing that it's, even though it's a granular form, meaning that it's not totally water soluble and it's in grains, like we see in magnesium, We've seen that by using this fertilizer, we're also applying magnesium, manganese, zinc, iron, copper, some sulfur, some boron, and some molybdenum. So this actually would be a good fertilizer for use in the Bahamas. And the cool thing about it is this here, because it's it has some sulfur in it. By applying it, you're also acidifying the soil a bit, right? Also the presence of the ammonia, the ammoniacal nitrogen, that is also going to acidify the soil a bit. This ammoniacal nitrogen and the urea nitrogen, that form is going to be available to the soil pretty much immediately, but with the urea is going to be limited, and over time some of it will be immediately available, but then the rest that is left, it's going to be available to the soil over time, right? And with fertilizer, you can do a one-time application, or you can break it into like do a set, like let's say a split application where you do it 2 times. Like, so if you're putting 10 teaspoons, you put 5 teaspoons in your first 2 or 3 weeks, and then the other 5 teaspoons in the next 6 weeks, at week 6. If you're gonna do like a water soluble fertilizer, what you'll have to do is, you'll mix according to the guidelines, and because you're applying it in the water, you have to be very careful that you're not watering and leaching out what you're providing. So you have to be mindful of that, and what will make sense to do is to apply it a little bit at a time. So you'll do a portion probably at 2 weeks, or well, in the first week you do a portion. Then, so let's say you do it on a Monday, then the rest of the week it's normal water. Then you probably do it whether a week after or 2 weeks after, where you're adding some fertilizer to the water. This is, I'm talking for the backyard gardener, but the same concept for a small farmer or even a large farmer. Because you're mixing it into the water, you have to be careful that you're not applying the water but flushing it out of the soil. So we have to be mindful of that. Okay, so let's have a look at some of the pests. So these are some of the leaf pests. So at the top here, we have tortoise beetle, armyworm, mites. Mites is not an insect. We have fungal spotters, and we have white flies, right? So in terms of these pests, so eggs, nymphs, larvae, or the fungal signs will either be on one or both sides of the leaf. So like the white fly, for example, they tend to be on the underside of the leaf. So you lift the leaves over, you see patches of the white flies. You may also see some leaf curling and yellow color. That could be an indication of virus, could be an indication of some kind of deficiency as well. Short holes like these here, frass, which is insect poop, chewed edging, evidence that the leaf has been chewed on. So some of the insects observed, they may also be vectors of virus, and vectors mean they carry the virus. And those include white flies, aphids, thrips. Using a systemic insecticide that targets the sweet potato weevil, which we'll speak about later on, that should adequately manage those pests. It's always a good idea to use clean planting material. That's one of the first steps for virus management in particular. So abundance of the leaf pest population, it may also be related to weather or environmental conditions. So for example, if the time is really dry, you're expecting to have mites, right? And you can deal with this by using an acaricide at the labor rate. At the cooler time of year, you tend to have a lot of the armyworms or caterpillars, feeding on the leaves. And if you leave them unchecked, they could literally just eat off all the top of the plants. And the top of the plants is necessary because those leaves are harvesting sunlight, converting it to energy. And that energy now is basically producing all that leaves and roots for the plant, right? And you can use a systemic or a contact according to labor rate to treat with those. So you might have to go in and do a knockdown for these caterpillar if they do show up. Let's go up to fungi. Just as a brief thing on there. So there's black rot. It can be serious on young and adult plants, and wounding increases how the plant is affected. And it can be spread by roots, soil, plants after harvest, sweet potato weevils, and rodents, right? So this is not present in all the countries in the region, or it has not been documented in all the region, but it is present in some places. So leaf spots, leaf rust, those are also, those might also be present in the field. And your cultural control. So your cultural control has to do with sanitation, the selection of the land, the selection of the plants that you plant, managing the weed. Well, weed management can be separate, but it has to do with those activities you do, your vine turning, all of those are what you would normally do to create that environment that allows for good management of any problems that might come up. Even creating a ridge, like preparing the soil by creating a ridge, that's a cultural control, especially in areas where you have a lot of clay and the soil doesn't drain well, right? So when they make ridges in those areas, what it is doing is it's increasing the height of the soil and allowing for, like, when it rains, that there's an area at the top of that ridge where the water is just draining off quickly. It's speeding up the drainage. So even that can be a cultural control, right? Rust here, as in on the right here, they tend to be of minor significance. And leaf spots as well, they don't always warrant any application of any chemicals unless you have a high damage. So you want to use clean, healthy, pathogen-free material. This is part of cultural control, sanitation, remove infecting plants. This is another term for this is roguing. Don't be afraid to rogue. If you see something that's off, it might be there being a source of the problem to spread to the other plants. So, don't be afraid to rogue. Disinfect your tools, because it's not good to be cutting here and then you might be cutting a infected plant and spreading whatever with virus or so to another plant. Managing the weed rotation. I know land space is but this is important that we rotate our crop. Solarization I mentioned, which is what you cover the soil and allow it to heat up to high temperatures, and chemical control. So you can use like oil sprays. This is good for the mites, but that would be fungus. But the trilogy can be used for mites as well. Okay, let's look at viruses a bit. With COVID in play, plants also have viruses. Their genetic code, either DNA or RNA, surrounded by a protein coat. It cannot replicate unless it's in the host, right? So viruses in sweet potato, they need a plant host to replicate, make copies of themselves and spread out. And they're transmitted to plants from parent plants through either fertilization or propagation or wounding. So wounding is something we should pay attention, as I mentioned, about the disinfecting the tools between using on a infected plant and moving to somewhere else, right? And the virus must be at the site of the injury in terms of this wound transmission. So some of the viruses in the present in the region that's documented. So we only have information for Jamaica and Barbados. And these are some of the viruses here. And we see that they're vectored or they're transmitted by either aphids or white flies. Some of the symptoms that we see, leaf curling. It can even affect tubers as well. So this kind of scratchy look on the, or etches on tubers that could be as a result of virus as well. This type of spotting as if it's like a color, like it's kind of bleeding like. So these ring spot like colors and these kind of chlorotic spotches here, and these would be some kind of discoloration, like a mottle kind of thing there. So some of these symptoms for virus. And another complication for viruses is that some of them are symptomless, like the leaves look perfectly healthy and so, but it may be a virus. It may have a virus, right? And the plant just doesn't show any kind of any symptoms like that. Virus control using virus tested planting material, that's often associated with tissue culture that does virus indexing, propagation, weed control, planting dates. Before planting, remove all the wild Ipomea species, which would be like a morning glory, and you use healthy planting material. Normally, if like your country has a tissue culture system set up where you can get access to clean planting material or virus-free index tissue culture planting material. After harvest, you remove promptly old harvested vines and burn or compost, and you harvest promptly. Don't allow the discarded roots to sprout. You can collect and feed to livestock, or you bury them or burn them, or take them to the dump, or put them in a bag, let it be solarized, and then you can dispose of. If you have resistant varieties, use those if they're available. So in the tissue culture lab, to get those virus-free planting material, they use the tip where you have the either meristem culture or microshoot tip culture, and they basically sterilize here this tip here, and they peel it back to the microscopic portion and put those in media and allow those to grow. It will be a slow growth, and then they'll have to, when it gets to a reasonable plant height, they'll plant that out in the media, in suitable growing media, and they'll have to weed and harden it because it's coming from this sterile environment and it's basically fragile, so you have to harden it a bit, and then they're able to get that out and plant outside, right? General disease controls. Because most of the time sweet potato is vegetatively propagated, we have to be mindful that we need to select healthy disease-free planting stock, have to be monitoring, keeping the place clean, don't be afraid to row, rotate where you grow, and prevention is key. And this applies also for insect and other pests too. So let's look at root pests. So we have here the infamous Xylus, which is at the top here, Xylus formicarius. It's one of the weevils. It's also called the sweet potato weevil. It is a major pest of sweet potato globally, right? And it affects the root. And the thing about it is that you can have a root and it looks perfectly fine, but then when you cut it, it has all these little larvae inside, and it's not edible because they make a mess when they get in, because the taste is horrible when you peel. Nematodes. So this one in particular is a root knot nematode. This is one of the only visible ones we can see visible. So these galls here, they tend to be associated with the female being present in the plant tissue, white grubs, and rats. Rats are not rodents, right? So just some pests affecting the root in CARICOM countries. So we have our weevil, sweet potato weevil, as you can see. It's pretty much in all the Caribbean countries. We have the West Indian sweet potato weevil, Eusepes, similar in behavior to the Scilus, or white grubs. We have rat. Also have nematodes. We have lesion, reniform, root knot. Okay, so the sweet potato weevil, this is the adult. This is the stage before it gets the color. So it's just emerged out of the pupal case, and it's gonna get its color soon. And what you're seeing here is, so this is a soil with some cracks in it. And these are the adults hiding, they're hanging out in the cracks. So a good thing to do in such areas is to, what a practice for sweet potato is to, after a while, you keep on healing up the area, right? And you don't allow for any cracks. So that means don't allow the soil to get too dry, because the adults, they don't dig well, but they'll find a way to go in cracks if they're available and eventually get to the roots, the roots. Right, so this is just in the tuber, how the damage can look. So this is on the outside, so you normally see like little pin pricks or pinholes. And then when you cut, this is a very bad one, like a bad infection here. So you even seen some larvae inside, and you seen the evidence of where they were tunneling. So they're going all around in the tuber. So a close-up of one of the larvae, and as you see, there's this discoloration after feeding, and this is the pupa. So from this stage to this stage, and then to the adult stage, right? And before that, there's the egg. So integrated pest management or integrated crop management interventions for Scilus formicarius. Cultural practices include those that help to prevent entry of pests or reduce the number of weevils, right? So clean planting material, removing your alternate hosts, field sanitation, ensure you have enough water, right? This prevents soil from cracking too much. Molding of the plants to reduce cracking. Harvest as soon as possible. So if you get a 4-month variety, harvest at 4 months, right? Don't leave it there long, because it's just going to be there and eventually they'll find a way to get in. Crop rotation, use varieties with lower susceptibility where available and possible, and use pheromones. So these low susceptible varieties, some of them, some of the varieties, they have immunity or a high level of resistance. Well, you don't have those that have a high level of resistance so far, but what we've noticed is that some varieties, they tend to have a low or moderate level of resistance. And some of them, this is simply because, the roots, they go down a bit deeper, so the weevil doesn't get there in time to affect them, right? Or some of them, they mature quickly. So let's say you would have treated the area, and by the time the weevils get there, they're already mature and you already harvest them. So, they escape that damage from the weevil, right? So because you can harvest early. Other intercropping, sweet potato with yam or cowpeas or ginger, legumes too. Rice, well, rice might not be applicable for some areas in the region. But this will be helpful for weevils. as I mentioned about using deep-rooted cultivars. So when the temperature is high, what we find is that the weevil can, instead of taking 30 days for the life cycle, it can go down to 20 days, so because of the temperature, it's gonna speed up the development time, right? So look, choosing what time of year you grow based on when you expect those continued high temperatures is something to look at. So looking at wet season versus dry season planting. You can do apply Beauveria bassiana if it's available locally to your root zone. Using soil amendment, neem cake, that can help to reduce damage. Actually doing some studies on that currently. Using sterile insect technique, which is basically rearing the larvae, doing a radiation treatment on them, and then letting it change to adults and releasing those adult males so that they go mate with the females. And by mating with the female, even though they lay eggs, the eggs they won't hatch, or if they hatch, they won't develop. So it's a sterile technique. The male annihilation technique is pretty much basically trapping the males. So you just have females out there, you're getting rid of, you trapping and killing the males. So that has been used in Japan, and successful eradication was confirmed for one island in 2020. But it's a very expensive endeavor, and they did indicate the limitations of that. And to reproduce it somewhere else, it will take a lot of resources, money, time, and a lot of a lot of adults that you'll have to rear to do it. But I think that's a good news. But for us to get to that stage, we'll need to do a lot of work.
So I actually have a little visitor. This is Silas formicarius, and it's here because I just opened a pheromone lure that attracts it, right? And that's one of the reasons I am wearing the gloves as well, because this is very potent, and getting it in contact with your skin, you'll be attracting these beetles to follow you out of the field. So I'm gonna go ahead and drop my visitor in here because I'm not interested in him. So what I'll be demonstrating is the pheromone trap. So this is the lower sector. This has the pheromone, and so you basically you can add it to the top here. So basically what we did is we got a bottle, and you could use any bottle here. Attach the pheromone to the bottle here, and inside you'll notice that there's some water here, and it's looking a bit soggy. What we did was we added some soap to the water, and what the soap will do is break the tension for the water. So if the soap is not there, what the insect will do is just land on the water and basically fly away. So just added a little bit of dishwashing liquid to some water, and that's your trap. What you'll also notice as well is that, so again, there's another visitor. What you'll also notice as well is that there are windows on the trap. So in this case, I didn't put the windows up here because I want, when it's raining, I don't want too much water to be getting in. So the idea here is that the water will go off and not a lot will blow into the windows.
Right, so basically you can use bottle, you cut the windows, you want to cut the windows below below that cone area there, because you want the water, you don't want to encourage a lot of water getting into the window. So the rain, instead of, if you put it at the top, you might be getting a lot of rain getting into the water and eventually flooding out the washing out whatever is in the container below, or filling it too quickly. Right, so you want to do that, and you can set up your pheromone trap like that. You also have commercial traps available. This is a modified trap. So this person is using a bucket. You're using soap, water, dishwashing liquid, adding a little dishwashing liquid to water, and that's it, plus the pheromone at the top, pheromone lower. And what the soap is doing, so if you just have the water alone, the insect will just land on the water, and they will be able to fly away, right? But having the soap, it breaks the surface tension of the water, so the insect will actually enter and will eventually drown, because the presence of the soap allows it to go below the water line, as opposed to staying on top if the soap is not present. Some places they also have the pheromone lure in the trap, but at the bottom or or all over the trap, what they'll do is spray or apply Beauveria or some kind of other entomopathogenic fungi. And once the insect lands there, it's going to be contaminated or infected with that fungi. And then when it flies away and interacts with the other insects, it spreads that fungus to them, and essentially kills the female and all of that it comes in contact with. Or even when it lands on the plant, it might leave some on the plant, and depending on how long that fungus is viable on the plant, then when the other insects land there, they'll be infected as well. And so you can use the traps to either monitor, or you can do a mass trapping approach, where you basically, because this pheromone in particular is a sex pheromone, and what it does is attracts the males. So by putting traps all over the place in a mass trapping technique, you're eliminating all the males in the area. So you have the funnel trap as well from the Taiwanese. This is in St. Kitts. This is also a variation, someone use a normal 1.5 liter bottle and cut the top and turn it upside down to create that funnel effect. This other trap here, where they just cut this another bottle here. You need 1 to 2 traps within 1 hectare. For the height of the trap, you want to put it either on the ground or just above the canopy of the sweet potato. So the sweet potato weevil, they fly, but they don't fly far, and they have a tendency, when the pheromone is present, they have a tendency to go up on the leaf, and you'll see them with their antenna, and they're doing a movement, and they're trying to pick up where it is to go and find it. So that's, having it on just above the canopy is helpful. Right, just some other ways how people set up the traps in the field as well. Right, so there's West Indian sweet potato weevil, Eusepes. Similar life cycle as I indicated. They're very hard to see. They're not as easy to see as the sweet potato weevil, because they resemble soil particles. but similar in concept, but we don't have a pheromone for this weevil, right? So this is damage versus, well, yeah, both of those damage and compared to healthy stem. So this is caused by the Eusebes in St. Vincent. And in terms of the tubers, you see how it damages the tubers, right? Compared to the marketable tubers here. Similar to the management is similar to silos. You remove and destroy the residues from the harvest. You do weed management. You sanitize the area. Remove your alternate hosts. Use the tip of the vine for cutting. Treat the plant material and the soil with available insecticide. You scout the field. You scout the field for damage, right? Grubs, so these are C-shape and they tend to browse against the tubers, causing mainly surface damage, but sometimes they go deep, so you see in there. We have different species, and all of them we just call white grub because of this C-shape, typical C-shape, and different ones in different Caribbean countries. So they lay the eggs in the soil, and they do like a surface damage to the tubers, and sometimes the fully grown larva will penetrate the soil and the pupae in there, right? And this is adult in this case for Phylophaga. So by managing Silas and or Eusebes, you usually take care of this. What happens as well is when you till the soil at the beginning, this will also, if these are present, these will also, because you're turning over the soil, these will also come to the forefront. And we know we have some birds that sometimes you see the birds going in the field and picking up. Yeah, this is one of them that they'll pick up because these are exposed when we turn over the soil. Removing weeds also will be a good thing to do, and treating the soil. We don't have any pheromone for this. You can use light traps. A lot of the times it's not necessary, but you know, you can use. Rats. Some of you who grow sweet potato might be familiar with this damage. Keeping the weeds down is a good approach for here. If removing a lot of the residues or the mulch layers is good, and you know, do your traps and your baits around the field. Nematodes, they're microscopic, so we can't really see them with the naked eyes. When they're damaged, when they damage the plants, we'll see the plants being stunted, so they're short, so they should be, you know, bigger than that, but they're short for the age sometimes, and the leaves become yellow and wilted. And you might have corky tissue as in the spongy, or you'll have galls like I showed you earlier, and you'll have some cracking. So rotating with peanut is a good option if you have lesion nematode. Chicken manure is good. Also, manure also helps to acidify the soil as well. So this nematode, it can be spread by transporting the plant root soil, and it also can be transported in surface or irrigation water. Right, so just a microscope for the lesion nematode. The root knot nematode, right? We'll see the damage. If you're actually seeing damage, then you know you have a high population, right? The rotation sequence is very useful for helping this. This is spread as well with transporting soil roots, and or it can be spread by water, right? If you have drought, you're going to see a lot of damage during drought time. Chicken manure is a good option for this. You can also have trap crops like marigold and crotalaria if you have crotalaria present in the countries. A good way to tell if you have root knot present is you put some tomato plants there and look for galls on them. Tomatoes tend to be very susceptible to nematodes. So that's a way to, if you don't have any lab services, you can plant a couple tomato plants and check the roots. And if you have these present, then you know you have to do some treatment for nematodes. Right, so these are some of the signs. So we have galls, that's normally indicates the presence of a female. So here I cut this root and you're seeing the female present here. So this is the mouth part and this is the abdomen area, and there's an egg sac. So why it has that ball shape, it's full of eggs, right? So it's a cyst-like. So we're looking for, but when we see in these galls, it's the females that we're seeing because it pushes out the right. So this is how the damage might look. If you look closer here, you're seeing a lot of small bumps or a lot of raised area. This is also there are two females here and their mouth parts in the tissue here, and as they damage, they cause discoloration as well. So you see the two shiny round things, and this is a close-up there. Management using clean planting material. You're cutting the slips above the soil surface, so go to the tip of the slip. Following is as in leaving the area without anything for a while. Soil solarization, rotation. For the chemical control, you have some nematicides that are on the market, but they have to be tested. Before, in the region, we were reliant on carbofuran and oxymil, which is, oxymil is the same thing as the active ingredient for Vidate, but it's not allowed in a lot of countries, right? In terms of Rotterdam Convention and stuff. And it's very broad spectrum. By applying it in the soil, you're killing good, bad, ugly, in between. And so this is something that we'll have to address as well, because we don't have much nematicides available, not only available for use in the region or even in other places as well, but we do have some potential candidate, and it's a matter of testing them for how effectively they work. But not to rule out as a direct in, which is the active ingredient found in neem. I mentioned earlier about the boron deficiency, and especially for alkaline soil, this is something we have to look out for. So this is some of the damage that we see when you have a boron deficiency. And this is also damage, how boron deficiency looks as well on tubers. So when do we harvest, right? The time between planting and harvesting is always a good measure. So when you get the material, they tell you it's a 4-month one or a 3-month one or 5 months or 6 months, you gauge the time when you plant and you go back at whatever time they said. So you base on time. That's the safest one. A common telltale sign is that you look the mature leaves, and the mature leaves will be the leaves that are closer to the bottom of the vine. Those will turn yellow and they begin to fall. Other way to look at for maturity is the root size. So they're going to be bulked up rather than, they're going to be bigger rather than small. Or you look for the presence of white fluid or exudate when you cut the root. So you're seeing some white exudate here. So white means it's ready, and black or gray or greenish means not yet. And you want to harvest as soon as the tubers are mature. And don't try to do where you harvest some now and cover back the root and go. Try to take out everything at the same time, because to lessen the damage of the weevil and fungal problems and so forth. So we've harvested, now what, right? We must be careful not to damage the tubers. And it's so easy to scratch the sweet potato. So you try to wash them as soon as possible after harvest. You can wash with water only first. And then you can do a 100 parts per million solution. So a rough example, if you use a 5-gallon bucket, you fill that up, and then you can add about 3 tablespoons of a 6% bleach solution to the bucket, and you wash those. So after you wash them, you air dry and leave them to cure at room temperature for 3 to 5 days. Try not to leave it in a high temperature. Then you store the roots as necessary and you prepare and eat. So recap: cultural control, crop rotation, sanitation, site selection, use of insect and disease-free planting material, very important. Weed management, following the land for a few seasons, using your pheromone trap to monitor insect populations or to do mass trapping, and your chemical control. Thank you, and God bless you. I know it was a bit long, but hope you enjoyed, and the floor is now open for questions. Well, I'm gonna hand over back to Dr. Bridgewater, because we're gonna do some poll again for the post.
So thank you very much, Dr. Annika, for such an extensive and very informative session. I think that most of us will agree that we definitely learned some new things today. So as promised, we said that we will get you back to the polls to find out if you have learned additional information, more information than when you started with the polls. So we, as stated before, your polls are located on the icon at the base of your screen. If you can click on that poll, you'll be able to see the questions that were answered before. And we would just want to get some information with regards if you are more knowledgeable than when we began this webinar. So in the interim, while we are getting those polls back up to you, we are just going to post some of the questions and answers, some of the questions that were asked by some of the participants, and we will refer those questions to Dr. Minott, who will be able to give you a response. As well, many of you were asking about whether this presentation is going to be available, and yes, it is our intent to have it available on the CARDI's YouTube channel. So we are going to just ask you to keep checking because it will be posted soon. So Dr. Minott, in the interim, until we get the polls sorted out, I'm just going to pose question 1 to you, which is coming from Ms. Angie M. And she's asking, which pesticides are used to treat the cuttings, and are these natural pesticides?
Right. So there are natural pesticides that you can use to treat the cuttings. So you can use neem oil. This is on the insecticide end. You can use neem oil, do a neem oil and water solution. You can also do use Beauveria bassiana solution as well. For the Beauveria, you'd have to treat for at least 30 minutes. So you'll kind of soak the planting material in that. For the fungicide aspect though, you might have limited options for the natural pesticide, but there are some oils that act as fungicide, and you might have some oils available that may have that fungicide and insecticide action. So those are possibilities. What would come to mind? Well, and it also depends on if it's available in your country as well. But yeah, you have natural pesticides that you can use, and you'll have to get an oil that has the fungicidal activity as well.
Okay, so thank you, Angie, for that question, and thank you, Dr. Minott, for your response. We now move on to question 2. Someone is asking, what is tissue culture plantlets?
So the tissue culture plantlets, those are produced in a sterile environment. What they do is so you'll select the slips in the field and basically use the nodes, cut it down to the nodes, and you'll plant them in a growing media in the lab. It's completely sterile, so it's free of any kind of germs and so forth. You'll also sometimes they grow it in a media that's treated with some kind of antibiotic or so, and you allow that tissue to grow out into a plantlet. And because of how you treat it with it, it's usually free of viruses, any kind of fungus, and also it will be free of insects as well, because you're basically growing it in this sterile environment. So this procedure is normally used for moving a lot of planting materials from country to country. Like, for example, people who grow bananas, and let's say you have a nematode problem in your country and you want to do like a large planting of bananas, you wouldn't really want to take suckers from your field, but you're able to get it, like those plantlets from whether they're imported or from a certified lab in the country. So it's a technique that a lot of that is used in terms of importing and moving plants from between countries or even within countries. It's also something that is used for citrus, and where you have like disease issues, and starting out with clean planting material is the way to go for those. So it's just plants that are produced in a sterile environment.
Thank you, Dr. Minott. So we would now move on to our third question, which is coming from Francesca, and she is asking, what is that sweet potato should be planted in and has not had sweet potato cultivation over 2 years? So she just wanted some clarity with regards to that area in the presentation.
Why is it that sweet potato should be? Oh, right.
planted in soil.
So, right. So I would have mentioned about rotation, right? So some of the times, those pathogens or those fungus that affect the sweet potato, they can stay around in the soil. So I plant the sweet potato here, I harvest it, but they can stay around in the soil as spores for a long time, right? And by waiting until 2 years, you're minimizing the chance of the sweet potato being infected with it again. So it's really from a rotation point of view. However, in the region, we do have limitations on land, space, access to land. So rotation can be in terms of time. But that if in an ideal world, you would wait until 2 years after planting. So this year, and I'll just give you a scenario. So this year, let's say I planted sweet potato January, harvested in April. It's like I don't plant back that area with sweet potato until 2 years from now. So that's what it's trying to say in a nutshell. But we have limitations, and that's because we have the pests present in the soil. So it's like it's decreasing your chances of having a good crop of sweet potato. And in some areas it's really bad. And you know, maybe it's a case where you just shouldn't grow sweet potato because you have to put so much inputs to correct the soil and to correct the environment to grow it. Well, it might not be for sweet potato, but in general, you know, maybe let's say you have a patch of land where you're always getting flooded, and there is, for example, like a Pythium or Phytophthora source somewhere near, like, and because you're always getting flooded, it makes no sense to grow tomato. So you maybe choose like a crop now that even if Phytophthora or Pythium is present, you still can grow something successfully. So sometimes it's also a matter of being wise and say, okay, this is just not good, not a good area to plant crop X. So that 2 years is just like a guideline. But it's not easy to do in the region.
Thank you, Dr. Minott. We now move on to question number 4. What do you mean with weathering as a cause of alkalinity?
So weathering is basically, okay, so like for example, the carbonate that you have in the Bahamas, so over time, it will weather, as in it will break down into different into the chemical elements. So leaching and the sun causes that deterioration in the original form of that. So it's now, so because of weathering, you're having smaller particles eventually, and the rock is breaking down into different particles and so forth. So it's just really like a chemical reaction based on environmental conditions and how the microbes, how those tiny particles are interacting with the soil and interacting with the chemicals within that environment. So in the soil, you know, it's basically a cycle. Sometimes microbes are holding some of these nutrients and sometimes they're releasing it. So in an acidic environment, you have a lot of free hydrogen, but in an alkaline environment, you have a lot of free hydroxide particles, sorry. You have a lot of free hydroxide. So the environment creates a situation where, because you see, weathering is on both sides. So weathering can cause acidity and weathering can cause alkalinity as well. So it's a matter of that parent material, how it's interacting with the chemicals, how it's interacting with the sunlight, how it's interacting with water, how it's interacting with the microbes.
So moving on to question 5, which is actually a combined question asking the similar thing. Is there any specific tool or instrument that can be used to test soil for pH status? And also in addition to that, how are persons able to test their soil pH?
So you do have either a pH meter or pH strips. Now, ideally, the best thing to do is to send the samples to a lab, because depending on what method is used, you might get an incorrect pH reading. So the best thing to do would be to send it to a lab, a soil lab for testing. But if you're not able to do that, in the meantime, you can use the pH strip or pH meter. And I don't know if possibly the agricultural agency in your countries, if they would at least offer that service to you. Because in their case, they could actually kind of do a calibration to make sure that the readings are okay. Yeah, so that for now, that could work, but the best place is to get it done at a lab.
And our next question is actually coming from Alicia Moses, and she's saying, "Hi, Doc. Can you tell us the varieties and the length of time before harvest, please? Also suggest pesticides and fertilizers for Barbados if possible."
Okay, so I really don't know what kind of pesticides, and I don't know what kind of pesticides, or can you put back the question so I make sure pesticides and fertilizers available for Barbados? But I do know that most countries, they have an all-purpose available. I know in Trinidad, their all-purpose is a 12-12-17 2, and the 2 is for magnesium, and I think they would have done that based on their soil. They would have done a soil survey. So most countries, they have a general idea of what's going on with the soil. And oh yes, to add to that pH part as well, so most countries, they would have done surveys, so you can get an idea of what the pH is expected to be in the areas in the country. So sometimes too, you can talk with your extension agent if available, or your agricultural person, and they can give you an idea of, the soil type, if it's, sandy, if it's clay, and what kind of pH to expect. But they tend to, you can get an idea of it as well if you don't have access to that. But the pesticide and fertilizers available, the extension agent would be a good place. And sometimes you can check with the agro shop as well and see what they have. For the varieties, off the bat, I can't tell you the varieties. I do know that there's some CARDI does have some varieties available in Barbados, because I do have a germplasm. I don't remember the names of some of them, but they can tell you, they can give you an idea. So like, for example, in the Cayman Islands, most of the varieties I have, they are mature at 4 months. So we don't have any early varieties. And there are a few that you can get a good yield at 4 months, but if you wait until 5 months, it's really better. So there are more 5-month varieties. But I wouldn't be able to tell you, but we could find out because we do have an office in Barbados and let you know.
Thank you so much, Dr. Minott. Here we have our 7th question. How do you control the sweet potato weevil? Info on the pheromone, and are pheromones available for purchase in farming stores? This is actually a combined question.
So pheromones, depending on the country, yes, the pheromones will be available in the farming stores. I know most of the times though, they tend to be in the government stores if your country have government stores. But some farm stores do bring them in because they know that you have sweet potato, you have a lot of sweet potato farmers there. So, but some countries it's available and some it's not. So it's more available in some countries than others. But it is available online. I think there are suppliers in the US. Possibly you might have suppliers in Trinidad too, but I don't know in terms of getting that to you in terms of what country you're from. For the weevil, so basically you can use a pheromone to monitor for weevil populations and then use that to determine when to spray. And you want to try to use a systemic insecticide that will penetrate that will be translocated in the plant, so that even if any larva is inside the root, it will be able to kill that larva. And you want to try to do that early in the cycle. So let's say you plant and starting out with land that you didn't have sweet potato, you should the weevils, if you didn't have sweet potato and you don't have like a lot of morning glory around, you won't start seeing the sweet potato weevil coming in until later, as opposed to if I literally just harvested sweet potato and go and plant back, I'm going to immediately see weevil around. So that means I'll have to start my chemical control early in the game, right? As opposed to if it wasn't sweet potato, then it might probably take like you might start seeing them pop up in the pheromone trap a month or so after. So yeah, prevention is a good control option. And while you're growing, you're healing molding up the area, keeping the area moist to avoid cracks, and have your pheromone for monitoring. You can put a lot of traps in the field too, to draw in all the males so that the females don't get a lesser chance to mate and lay eggs on the tuber.
Great. So our next question is coming from Elizabeth Mohammed Ali. Thanks, Elizabeth. She said, I saw here in Trinidad, we are using molasses at the bottom of the traps. So possibly she's asking, can molasses be used as an alternative to dishwashing liquid or soap water at the bottom of the traps?
I think I would need some clarification on that, because there's also a trap for the South American palm weevil of coconuts where they use molasses as part of a food bait. So that would be one of the ingredients for that. But I'm not familiar with using molasses in the trap with the silas. But I would imagine, so in the case of silas though, I would imagine that molasses would be a bit of an expensive input in the trap. And as opposed to the soap, a little dishwashing soap and water, and I mean that does the job. So as I said, I would need some clarification because there is another trap for another insect that uses molasses.
Great, thank you. And Franny Thorpe is actually asking, she's been using the same rodent control consistently. Eventually, it has been becoming non-effective. And she's asking how this can be addressed.
Okay, maybe you could look into and hopefully you'll have a different, maybe you could look into a different active ingredient. I'm not sure how available those are in your country, but you could probably try a different active ingredient and see if that helps, or even a different kind of trap as well. But I don't know if the garden shop can tell you what active ingredient is in the bait that you're using now, but you could try a different one to see. Because sometimes I know with the rodents as well, after a while it's like they kind of expecting, oh, this is not good for me, so they leave it alone. So maybe trying, shifting things up on them should maybe work.
Excellent. Now, at this time, our next question is, what is the ratio of water to bleach when washing potatoes? And that's coming from Rosetta. Thanks, Rosetta.
Right, so I'm gonna go look back on that screen there, right? So 100 parts per million. So as I said, so it's if you have a 5-gallon bucket, you just use 3 tablespoons of the bleach. And for the bleach as well, try to get Clorox or one of them that tells you the percentage of the sodium hypochlorite. I know sometimes you have bleach on the market, but it doesn't tell you how much percent. So like if you buy one that says 6%, you just use about 3 tablespoons of that, and you add that to a 5-gallon bucket, and yeah.
And as we move on to our next question, the question is, what is the proper way to store harvested tubers to extend the shelf life?
So curing, as I said, is a good start, right? So you're going to, so in terms of what I outlined, let me go back to there. So if once you cure it at first and then store, that's a good start. So you wash the tubers, you let them air dry. And it's a good idea not to pile them on top of each other, because sometimes that is causing, wounding and all of that. So you kind of, either you try to do a single layer and or probably a double layer, leave those to dry. Then now you cure for the 3 to 5 days. Then you can put them in like cardboard boxes or something breathable and leave for storage. You can chill, you can put a chill room, or you can leave them out in a cool area. Low oxygen is good, and just avoid any kind of bruising. One sec.
So Dr. Minott, I don't know if you're finished with that question. We probably
Yes, I am.
Okay, so Mr. Dorset is asking, can I plant cassava in a field that I recently harvested sweet potato from? I think probably not, but I want to be sure. Thank you. Thank you, Brian.
Okay, hold on. Sorry. in a field that I recently. Okay, so because you said, can I, can I plant cassava in a field? Yeah, you can. So cassava and sweet potato, they, thank God, they don't have the same pests. And so you can. So there, it's two different pest complex there. It's two different pest complexes, so you definitely can do that. And most of the times cassava is going to take up the area for at least 9 to 12 months. So that's a good long time without any sweet potato there. But you're going to have to make sure that you're fertilizing properly, because both of them, well, cassava not so much, it doesn't take up a lot like how the sweet potato does, but the sweet potato does take a lot from the soil. So you have to make sure that you're fertilizing properly, because both of them can take from the soil. So apart from that, Miss Beckles, right. So in terms of this, the storage routes going all over the place, what could happen as well is that you, let's say you you thought that you removed all the sweet potato from the area, because there's a tuber somewhere down. Because so basically I could have a tuber here and it sends out a pencil root, and then it decides to swell up here, but there's no evidence that there's a sweet potato on top. So I went ahead, I removed the top portion here, and I'm like, okay, I don't have any more sweet potato in the field. And then a couple weeks after, I see a sprout coming here. So I mean, because of that, now you know you might be, if you're planting sweet potato, you're encouraging a problem on your end, because you want to try to keep the area free of sweet potato for a while, but because you're not because you didn't do that, no, it's not really free of sweet potato, right? But those are pretty much the reasons why. But from a disease management or a pest management viewpoint, it's not ideal. But I know sometimes it's good too that, you know, for some people, like they probably something damaged the sweet potato here and they thought they lost the variety and stuff, and then lo and behold, the seed sprout up back another time. So, but on a commercial level, it makes sense to just do it from where you just go and harvest where you plant it.
Great. So guys, that was actually our last question. It has really been a very long and very interesting morning. And I know that everyone is very interested in this topic, and you are trying to get in as much questions as you can. So what is going to happen? We are just going to share with you, coming closer to the end, possibly an email address where you can address your questions, and someone will definitely respond to you. So it's not that we are not going to answer your questions, but we would like to end the session at this time. And we just want to bring up our poll once again, because some of you were unable to complete the poll. So we're going to bring up the poll along with some of the results as well.
You want to share how we'll answer the questions? Will we email them, or just how we gonna?
Yes, we are going to be emailing, sharing an email at the end so that you can possibly send your questions. And we also have the questions saved as well. So we would see how we can possibly incorporate it in the YouTube presentation as well, so that your questions can be answered. So now we are just going to share the results. So one of the first things is in terms of question 1, most of you answered that you will tell by size. You have quite a large number of persons who said the mature tubers will release a white fluid and exudate when cut. So in response to that question, how do I tell if sweet potato storage roots are ready for harvest? Dr. Annika, would you just like to say which one applies?
Yeah, so both the size and the white fluid, those are the two answers there. So those would be the two correct answers.
Our second poll question, weeds compete with crops for space, light, nutrients, water, all of the above except light, or none of the above. What is the correct answer?
Yeah, all of the above would have been the correct answer.
Right, and we had 86% of our participants actually getting that question quite right. Number 3, in terms of cultural practices that are useful in managing pests and disease in sweet potatoes, is it chemicals, crop rotation, using clean, healthy planting material to establish crops, sanitation, keeping the area planted as clean as possible, all of the above, or only B, C, and D only?
Right, so, well, only B, C, and D. I said cultural, but yeah. I understand why they would have said all of the above, but yes, in this case.
Our weeds can be an alternative host for viruses, insects, nematodes, fungi, and bacteria. Is this true or false? We had an overwhelming response.
True.
Excellent. And what are some of the examples of pests affecting sweet potato roots? We have weevils, rats, nematodes, whiteflies, caterpillars, and grubs.
Okay, so weevils, rats, nematodes, grubs. I can make an exception for caterpillars because in Trinidad you do have the stem borer. But whiteflies would definitely be sweet potato leaves.
Yeah. And what are some of the examples of pests affecting leaves of sweet potato? Is it the weevils, rats, nematodes, whiteflies, caterpillars?
So the leaf one we are now?
Yes, correct.
Right, so it would be whiteflies, caterpillars. You do have the weevils, they do feed on the leaves a bit, but not majority. But rats and nematodes would not be for leaves.
And our last question, poll question, is that soil pH is more than 7. What should I do to the soil?
And that would be add an acidifying agent.
So once again, we would like to say thank you. It's been a really exciting morning into afternoon, and we hope that you learned a lot more and that you receive a lot more information than you had when you first started this presentation. So from CARDI and all our collaborating agencies, we would like to say thank you and stay tuned, because there will be soon coming and upcoming Roots and Tubers series training. So listen out. Thank you.
Right, so we will be answering everyone. So don't worry about not getting